Hello everyone, my name is Dr. Hafsa Abbas, and today, as part of this great annual event of World Cancer Research Day, the aim is to conduct a lecture for the general public titled An Insight into the Importance of Cancer Research. So just a gentle reminder, we are not associated nor affiliated with World Cancer Research Day organization. World Cancer Research Day organization was established in 2016, and it consists of over 10 world cancer research organizations, such as World Health Organization, WHO, Cancer Research UK, ASCO, EACR, amongst many others. However, at FST, we have projects that are faith related, art and community related, and also health. So in relation to the health, our main focus is within cancer research, and we also provide health articles. Hence why we are able to bridge a link between World Cancer Research Day and ourselves, because we understand the importance of cancer research and raising awareness, which is part of our aims. So I would like to initially thank World Cancer Research Day organization for establishing this annual event every 24th of September. I would like to thank the Divine, myself, family, friends within FST and outside of FST, and anyone who helped during my academic journey. So just a brief insight about myself. I initially began within the cancer research field in 2013. So humbly speaking, I've experience within patient care as well as cancer research within the laboratory. And within my research journey, I have been able to conduct research within the fields, write articles, and also conduct lectures on the field. I've also participated in international and national cancer conferences and have recently established a biannual medical journal which is catered for early cancer researchers like myself, with the aim of building abilities, confidence, and self-esteem through research and development. So how is this lecture catered for the general public and all learning needs? Well, firstly, there is factual information that are broken down in simple terms with supporting images and flow diagrams. So this is suitable for anyone who learns through images and picture formats. All information are fact-checked and full reference. So if there's any information and you wanted, okay, I want to read about this more, there is a reference there where you can refer to it and learn about it. Okay. Thirdly, there's an opportunity for you to pause and take small mini breaks. So this will help to absorb the information and process before continuing the lecture. So this is why this audio lecture is actually suitable for one-day workshops, or if you have a series of coffee mornings where you discuss about cancer, and then you can add this as part of your coffee mornings where you can pause the lecture, discuss, reflect, share experiences, and also understand the importance of cancer research. And for anyone who likes to learn through lecture notes or wants to make notes, there is free lecture notes in PDF format. Okay, so you may want to add additional notes during the lecture or during your discussion within your coffee mornings or with your focus groups and so forth. The audio lecture is recorded on YouTube. So if you live in a different time zone, or let's just say that you needed to pause the lecture because you had to do something, that's okay, because no matter where you are around the world, whether you're in the Far East, Europe, Africa, Asia, or any parts of America, you don't need to kind of like remain native at a fixed time. It's open for, anyone who can go onto our YouTube channel and be able to listen, learn, and share. So here we have an outline of the lecture. Firstly, we're going to discuss what is cancer and what are its main risk factors. Secondly, we'll then progress on to understand why cancer research is important. Okay. What type of cancer research are there? Thirdly, we will discuss what is World Cancer Research Day. Okay. What does the organization do? We will then progress by understanding the techniques or the method or how research works. So with medical research, what are the key steps involved? And we're going to be focusing on two main types of research within the cancer, qualitative and quantitative. So we're going to be able to define it, understand what it means, be able to describe it by referring to the different types of each category, and be able to provide examples for each. So for instance, you may come across this research data and say, okay, this is quantitative. Oh, right, this is qualitative. So you'll be able to tell the difference between both. And then last but not least, once the general public has been able to understand the importance of cancer research, the next step is, okay, how am I going to get involved? Okay, so you may want to volunteer for a cancer charity. You may want to fundraise for a cancer charity. You may want to be able to take part in the clinical trials to try and make a difference within cancer research. There's different ways in how any member within the general public can help make a difference. So what we're going to do, we're going to start off by first defining what is cancer and what are its main risk factors. So what is cancer? So cancer is a disease where there is a change within the genetic material, okay? And this is called a mutation. And when there is a mutation, what happens is the cancer cells start to grow, okay? The cancer cells start to grow, and it grows and it grows, and then the shape changes. So there's a big difference from a normal cell and a cancer cell, and also how it works. Okay. So as you can see in this diagram over here, you have the initial light pink, that's a normal cell. But when there's a change within the gene, you can see there's a change in its shape and color, and then it starts to grow. Yeah. So cancer can affect how the body responds. In other words, the immune system, because the immune system's role is to be able to combat any infection and diseases such as cancer. Okay, but if they do not respond appropriately, then the cancer will be able to grow and grow. Now, there is two different types of cancer. There's the benign tumor and there's the malignant tumor. The benign tumor, as you can see over here in the image, the cancer cells which are in pink, they are kind of clustered within an area. Okay, so they're not really grown or spread in different parts. So this is why we refer this as a benign tumor. In this second image here, we have the malignant tumor. So you can see there's continuous growth of these cancer cells that it's basically spread across the different layers and has been spread out. Okay, so where does it go? It affects the neighboring tissues. It can also affect the organs. How does it travel? So this is where we're going to discuss. One of the ways in how cancer cells travel after it grows from the primary sites is via the blood. So that's how it travels to other organs and tissues. So what is the blood? So this image presents red blood cells, white blood cells, plasma, and the platelets. The blood also consists of nutrients, hormones, and this just helps to ensure collectively that the organs and tissues are working well. The role of the white blood cells, they are part of the immune system. So when they see an infected cell or a bacteria or a virus, they try to remove it straight away. The platelets, if you've managed to, for instance, get a cut, what the platelets do is try to stop excessive blood loss. Okay, because when you lose a lot of blood, you can potentially become anemic. And this is why the platelets, what they do, they clot together and they stop the blood loss from occurring. Okay, so this is why you have like a scab or a mesh at the end. The red blood cells, they carry oxygen around the body. Okay, and the plasma is what carries the nutrients, the hormones, and the blood cells collectively. The other way in how cancer cells travel is via the lymph. So the lymph is clear and colorless fluid, okay, and it contains the immune cells such as the T cells, the white blood cells, to help protect the body from infection. So as you can see in this image over here, the lymph may look green, okay, however, in real life, it's actually clear and colorless. Okay. So in this image, you can see you've got the blood vessels. So the venules are small veins. Arterioles are small arteries. So they try to carry blood depending on the level of oxygen. And also the lymphatic vessel, that's what carries the lymph. So what causes cancer? So what causes actual cancer? And one of the ways is through the family. How? It's because it can be passed through the family. So if let's just say uh for there's a change within the gene, a mutation in a part of the gene, let's just say APC, what that does, that can increase the risk of colon cancer. So that's the cancer of the intestines. If there is a change in a gene or a mutation, let's just say in HER2 or BRCA1 or 2, or there's a change within the estrogen, that's what leads to breast cancer, ovarian cancer. I mean, there's different types of genes, and that can, if one family member has that particular mutated gene, it can actually pass or increase the risk, which is why you have cancer screening. So within this image over here, you can see that when there is a normal gene, it produces a normal protein because the gene is what carries the information to make a protein. That's the link between a gene or a protein. So the gene carries the information that makes a normal protein. A normal protein, we need it for growth and repair. Okay, you can find different types of proteins within our body, in our hair, nails, in the blood, hormones, uh, you know, uh, muscle area, variety of different locations, or in the digestive system. So when we break down food, we need proteins to help to do that. So there's different aspects there. Now, when there is a mutated gene, where there's a change within the gene, that is what leads to an abnormal protein. Okay, so the protein won't work anymore. So what we have over here is a diagram of how it can increase the risk. So let's just say we have a mother and a father here. So the father carries a gene mutation. Okay, so it has the gene mutation, and the mother is not affected, so she doesn't have the mutation. So they've gone off and created their own family. They were able to get 2 girls and 2 boys. So on a basis of, because the father carries the gene mutation, there's a 50% chance, okay, that their children will be able to have the gene mutation because the gene gets passed on. Because half of our genes within our body is from our mother, and half of our genes from our of our body is from our father. So we've got half genes from the mother, half genes from the father. So this is why you can see there's one son that's not affected, one daughter is not affected, one daughter is affected, and also one son is affected. Another cause of cancer is lifestyle factors. And I think it's very important to be able to distinguish the difference between cause and risk. Because if one has for instance, smokes, or is overweight, or is alcoholic, has poor diet, poor physical activity, that does not necessarily mean that you will 100% get cancer. No. What it does suggest is that it increases the risk of cancer. So some of the reasons why there is a change in a gene is not necessarily because of family genetics. It could be other causes. So let's just say you've been smoking or drinking. So what they have is they've got some cancer-causing chemicals called carcinogens, and this is what increases the risk of mutations and cancer growth. Okay, so yes, lifestyle factors does increase the risk of cancer, which is why, for instance, the NHS, for those who come under the category of drinking alcohol, it's been recommended that they would have no more than 14 units of alcohol, and that's regardless whether they are male or female. Another cause of cancer or increase the risk of cancer is industrial chemicals and air pollution. So if you're working within an environment where you have strong toxic chemicals or utilize very strong fertilizers, or your factory releases a lot of industrial gas, it does not only just lead on to, for instance, global warming and change of weather and toxic in terms of the trees and nature itself. It's actually toxic for the individual who works with such environments because to be able to breathe in these chemicals. And this is why it's important that safety, where and how much um, exposure you have, how much chemical exposure you have, or radiation exposure you have, can make a difference. So the lower the exposure that you have towards these chemicals and radiation, the better. There are also biological factors, so infections that are caused by viruses, bacteria, and parasites. So all of these are considered as microbes, so you won't be able to see them on a naked eye, for instance, on a table or in the air and so forth. You would need to utilize an instrument called a microscope to be able to actually see them. Okay, and you will be amazed that there are different types, shapes, colors, formats, you name it. So, and I think it's important to state that not all fungi and bacteria and algae are actually cancer causing, okay? Because you have some bacteria that are good bacteria, and this is what able to make cheese, milk, yogurt. So the lactobacillus is another one example. Who likes mushrooms? If you like mushroom on your pizza, that's a fungi. Also, um, yeast. So how we make our bread, yeast is also example of a fungi. So there's not all are can cause cancer. There was some species that does. Okay, but in terms of viruses, there isn't really a good virus. A virus is a virus. It just causes symptoms. So one of the examples of the viruses that can lead on to um, cancer is hepatitis B and C. Okay, so hepatitis B and C viruses can cause liver cancer. So initially, it can cause an infection. Okay, followed by an infection, it can cause the cancer to undergo some form of texture like fibrosis, and then eventually liver cancer. Also, when you drink a lot of alcohol, for those who are under that category, that can cause cirrhosis. So that can stop your liver from functioning appropriately, and that can also increase the risk of liver cancer. Another type of virus that can cause cancer is human papillomavirus, HPV. So HPV, there's lots of different subtypes, but the main types of this virus that can cause cancer is 16 and 18. And with this type of viruses, that can be through, bonding or contact. It can be passed through bodily fluids, blood, injections, and so forth. And this is why the NHS conducts the smear tests for those who are eligible every few years, just to lower the risks, because the earlier you can find a cancer, the better. Now, as you can see within the images over here, this type of cancer normally affects women. Okay, so the cervix is found within the female reproductive system. So when you have a healthy cervix, okay, and then you have a cancerous cervix, so the cancer be able to spread outside the cervix, especially in the lining here, which is why you can find some bleeding and cancer tissues there. The food type of cancer is Kaposi sarcoma. So this is a cancer normally affects vascular tissue, so blood vessels. Okay, so it can commonly occur on the legs and different areas, and it's categorized by having these brownish purple noodles. So you can see over here, some are found on the feet, which is presented here. You can also see it's affecting the mouth. So those who have HIV are more at risk of having Kaposi's sarcoma. So this is soft tissue cancer. And if you can look in this image over here, how the HIV is able to affect the immune cells, so it can affect the T cells, and this is why the immune system is compromised or not working, because how the HIV targets these immune cells by being able to enter the white blood cell, change its genetic material, be able to grow its own small viruses, and then be able to release it. Okay, so this is very, very dangerous. The next step we're going to proceed on is, what is cancer research and why is it important? So what is cancer research? So cancer research, to break things down, is research finding out, investigating what cancer is. How do these cancer cells work? Because there's not one type of cancer, there's so many different types of cancers. And there isn't a single treatment that can treat all of them. So you have different types of treatments for different types of cancers. And then there's the question here is, okay, which treatment works better? Okay, how do these cancer cells work? Why isn't this treatment working on the cancer cell? Are there different ways these cancer cells able to grow? So this is why cancer researchers are always on the go in trying to find new treatments. Okay, because if a patient does not respond to a particular treatment, they be able to become more resistant, and this is how the cancer cells be able to grow. So this is why it's always being able to find and develop better treatments. So that's the difference between patient side and the laboratory bench, because with the patient side, you have contact with the patients, try to get them reassurance, answer any questions, reassure the family, find out how they're doing, how's the blood test, are the cancer cells going down, is the immune cells going up, are they responding? Generally, how are they responding to the treatment? Lots of scanning tests, and so forth. And when you're on the bench side within the laboratory, this is about, okay, how am I going to help this patient? Because what about if this patient is not responding to the cancer treatment or is not working effectively? Can they do some cancer clinical trials? We're trying to find the dose will it work effectively against that particular tumors, especially when, for instance, with brain cancer like glioblastoma, which is a very aggressive cancer, and it has very poor response in terms of for treatment. And this is why it's very important to conduct cancer research to understand how it works, how can it be diagnosed better, how can we have an early diagnosis, how can we be able to develop better treatments. And this does not always necessarily work on a laboratory bench. Sometimes you can understand how the patient feels or their experience is through conducting patient surveys. So finding out, okay, what dose have they been having? Have they been experiencing any side effects, symptoms? How is their quality of life? How is it changing? Is it going on a better or the latter? Um, uh, so finding their experiences there. Also, how they be able to, for the lifestyle factors, do they actually smoke? Do they drink? What do they eat? Do they eat healthy food? What type of healthy food? How much exercise that they do? So there's a variety of different types of surveys that one actually can do. In addition, you have monitoring the incidence and death rates. So this is a more of a study known as epidemiology. So what they do, they kind of monitor how much patients are being diagnosed each year. So diagnosed as in how many have been told that, okay, you've got cancer. Okay. How many have unfortunately passed away from cancer? Has this number increased? Has this number decreased? And it's just about finding out, okay, is this cancer charities, are they working effectively in raising awareness? Is the health promotional material being effective enough in raising awareness in terms of the screenings? Is it are there people aware of the symptoms so they'll be able to go to the GP much more sooner? Do they actually go to the screenings programs or do they not participate? And have they participated in the cancer screening? So there's a lot of questions here and there, and socioeconomic status, for instance. Do they live in an area where there is poor access to such health information? Do they live in a location where there's poverty there? Or because with different locations, even within the UK, there's a different response in terms of the incidence rate. For instance, you can find one region there's a higher incidence rate than another region, and it can be related to transportation, having specialist hospitals, and lots of questions asked. So an example one that we're going to present for a survey is here. So this is a National Cancer Patient Experience Survey of 2024, and they were recently published on the 17th of July 2025. And those who have been enrolled onto this survey were aged 16 years and above. And based on this survey, as you can see over here, there's a 50% response rate, and that 50% equals 64,055 people. Okay. And based on the survey, 8.94 gave an average rating care of out of 10 from the NHS. So they felt that their care on the NHS was great. Okay, so it's 8.94 out of 10. 87.6% said that they felt that how the care was organized, for in terms of the paperwork, administration, calls, contact, communication, they felt that was good. And then 90.4% said the whole team actually worked really well together. So these are some of the statistics there. And there is a lot other more details, such as the right level of support. 77.5% felt that it was, and 80.4% felt that they were more involved within the decisions of the treatment. So there's a lot of key facts over here. So this is an example in how data can be collected. And they could, it's not only in terms of the treatment in terms of in-house within the medical care, but also, for instance, getting enough emotional support at home. 33.5% said that they've had that kind of support after treatment. 64.9% felt that they were given enough information about the possibility of the cancer coming back. So let's just say they've been told they're cancer free, but there's a likelihood that it might come back if so and so happens. So there's a percentage there. So there's a lot of more information. So do visit ncpes.co.uk to see more detailed results on this. The second example that we have over here is monitoring number of cases over a period of time. So this is an example from the International Agency for Research on Cancer. So this is part of the World Health Organization that collects data from around the world about the incidence, so the number of cases that have been found, and also the number of cases that have been passed away, so mortality. And the number of cases are highlighted in red, and the number of deaths are highlighted in more of a navy blue black color. So there is a drastic difference from 2000 all the way to till 2050. So if we reflect in terms of the number of cases in 2000, there was 10.1 million. Okay, and then it doubles to 20 million in 2022. And based on these estimations, they have been considered that by 2050, there's approximately 35.3 million. So one would ask, okay, why would the number of cases increase? Why? I mean, what will trigger or increase the number of cases there? But if we actually compare the number of cases for cancer incidence and death, it is increased by approximately by 20% between 2022 to 2030. So even within the intervals, there is a significant difference there. So one would question as to why. Is it lifestyle factors? What could be the trigger into rise of this? Because there's a variety of different established cancer charities they're raising awareness, the NHS is raising awareness, the government is raising awareness, who is raising awareness? So one would actually ask, okay, what is the cause of this? So this is something one actually need to explore and think about. So from the 134 of the 183 countries in which the World Health Organizations have founded for this, is that cancer actually leads to premature deaths, particularly between 30 to 79 years. So despite the aging population is increasing, the premature deaths for those who are in the early adults all the way up to the elderly, cancer actually can lead to premature deaths. And this is why, or may explain, why there was an increase from 6.2 million in 2000 to 9.7 million in 2022. So cancer also imposes an economic burden. So it's not only just about the incidence rates over here, it will have a significant burden on the different healthcare systems across the world. And in the case of the UK, the NHS. So it's been estimated by WHO that there'll be $26 trillion that will be spent between 2020 and 2050 just for cancer alone. So if you could think about how, for instance, cardiovascular disease, heart disease is the biggest killer worldwide. And then you have cancer, but you've also got lung disease and many other diseases, but these are amongst the highest one. How that can significantly affect the healthcare system. So this is why it's very important that we are able to raise awareness of the different risk factors and to take heed. Example 3 is a laboratory result. So this research results was being published by a group of researchers called Kaliskan et al. So what they did, they created 12 new chemical products by joining together the amino acids with active agents of a chemical called cinnamic acid, which is extracted by plants. So they've made 12 new chemical products by joining together amino acids. Again, and amino acids, these are building blocks that make up the proteins. So the genes carries the genetic information to produce the protein. The amino acid is like the bricks that make up the protein based on the genetic information there. And have these products not only been made with the amino acids, but also the active agents that are being extracted from plants. These active agents are vitamins, hormones, steroids, sugars, amino acids, and other chemicals. So this is why the new chemical is called a conjugate, because they've joined amino acids with these active agents to make a new chemical product called a conjugate. So there's different conjugates there. So the new conjugates produced by Kaliskan et al. Were tested using 4 cancer cells. So this has been done within a laboratory. So the first type of image that we have over here is the MCF7, which is a breast cancer cell line. Okay, now the first bar. So let's look at one paragraph per one graph per one graph. The first graph over here with the MCF7 cell viability. So cell viability is the number of cancer cells that are alive, and they've done that in a percentage. Okay, so the first bar is the control. So the control means that they do not have any of the chemical, the conjugate. Then you have a solvent, which is something to compare it with. And then you have different concentrations of the conjugate. So you've got 1 micromolar, 5 micromolar, 25 micromolar, and 50 micromolar, and 100 micromolar. So these concentrations of the conjugates is the same in the different cancer cells that they've utilized. So you've got breast cancer, prostate cancer, colon cancer, and ovarian cancer. So they've kept the number of the concentration the same, different. So 1, 5, 25, 50, and 100. Okay, and they're comparing it without the new conjugate, i.e., the control. So what they did, they wanted to compare it with 5-fluorouracil, which is an approved chemotherapy that's already utilized in a variety of different cancer cells. And when you have the cinnamic acid alone, okay, without the conjugate, you will find that it's less effective in killing cancer cells. However, when you've got the conjugates, i.e. the 1 micromolar, 5 micromolar, 25 micromolar, 50 micromolar, and 100 micromolar, you can see that the number of cancer cells are being killed, okay, it's actually increasing. And this is why you have a smaller bar in each of the case. So based on these graphs, the response actually varies. So let's look much more closer. So you have the MCF cell viability. So you've got the control 100%. We've got the solvent, which is about approximately 90%. And then when you have the 1 micromolar of the conjugate, it decreased significantly to approximately 70%. And then when to 5 micromolar, there's a slight decrease there. And then with 25 micromolar, surprisingly, there is a decrease in comparison to the control, but it's slightly more than 5 micromolar. And then when you increase double the concentration to 50 micromolar and 100 micromolar, there's not much difference there between 50 micromolar and 100 micromolar, but you can see there's less than 60% cell viability. However, when we look at CaCO2 cell viability, the control, there's not much cancer cell death. With the solvent, there's a slight decrease, but it's not really a lot or significant. But suddenly here, when you have 1 micromolar of the conjugates, and then you start to increase the dose to 5, 25, 50, and 100, there is a significant difference compared to the control, which is the cinnamic acid alone. Okay, so there's approximately, let's just say, 100% for the control for CaCO2 cell viability. But as soon as you add 1 micromolar, there's approximately 30% in 1 micromolar. Then it starts to decrease to approximately 25, and then much lower. But similarly, there is not much difference between 50 micromolar and 100 micromolar. When we reflect onto the PC free cell viability, the response is lower. So you can see there's a decrease of cancer cells in 1 micromolar, 5 micromolar, 25 micromolar in comparison to the control. There's a slight increase in the 50 micromolar, but there's a huge difference between 50 micromolar and 100 micromolar in comparison to the other two cell lines that we discussed there. So there is about 20%, but the response was much more stronger with the CaCO2, because even with a lower dose, there was a much more cancer cells decreased at 1 micromolar comparison to the other two which we just done so far. And when we reflect in terms of the A2780 cell viability, what we can see over here, there is some significant decrease there, but it's more similar between 1 micromolar and 5 micromolar. The red is approximately, let's just say, about 62 or 65% cell viability. So the remainder are the number of cancer cells that were killed. Or when you increase the dose, it starts to decrease. So, but the one that has the highest response based on this data is CaCO2. The one with the least response, I would say, is the MCF7, based on the concentrations over here. So, but the in between is a PC3 and A2780. But collectively, what we can see is that concentration of the new conjugates, as it increases regardless of the type of cancer, there is a difference in comparison to the control and the solvent. And how or what causes this is because when you have the conjugates, it tries to kill the cancer cells by damaging the DNA, so they can't really grow. The next part which we're going to do is to understand, why is research in cancer important? So cancer research is mainly focused, especially across the world, and these are data from the World Health Organization and World Cancer Research Day. It's the leading cause of death. Okay, so after the heart disease, the coronary heart disease, the stroke, and other types, cancer is amongst the leading causes of death, and it's nearly caused 10 million deaths in 2020. So that's 1 in 6 people. The number of cases are increasing each year. So there's 24.1 million cases that have been estimated by 2030. The most common cancers are breast, lung, colon, rectum, and the prostate. So breast, it can affect males and females, but more in females. Main factors are genetics, hormones, lifestyle factors. Lungs, mainly lifestyle factors, such as especially with smoking and working within toxic chemical environments. Colon cancer, it could be genetics. It could be associated with the lifestyle, such as eating red meats, tomatoes. Also, if you have other conditions, polyps, that can also increase. So these are like abnormal growths within the intestines. The rectum, this is where the stools are being stored. So this is part of the digestive system. The prostate, so that can be related to lifestyle factors. It can also be associated with certain races that are more at risk than others. So there's a number of different factors there. Emotional impact. Why is researching cancer so important? Because it can affect everybody. It can affect you, can affect me, can affect anyone who knows somebody who has been diagnosed by cancer, knows somebody who's been passed away from cancer, or they themselves has been diagnosed with cancer, um, and have been cancer free or currently undergoing treatment. It can affect anybody. Okay, it can not only affect them, it can affect the families, it can affect the friends, the communities itself, if the community is close. It can affect their personal life, the quality of life. For instance, there's certain activities they may need to pause or change or find new hobbies so they can make use of the time. There's a life adjustments, so especially if you've got youngsters or have a particular job and so forth. And the different signs, the symptoms, also your response to the treatment, side effects, the status, for instance. Okay, I want to know how I'm doing. Is the treatment going well? Am I responding well? So it's always an emotional impact there, and it's natural. Economic impact, pressure, so that can why? Because the money spent within the actual healthcare systems, the premature deaths, the cost of the treatment, the clinical trials, the cancer research studies itself, um, the care, transport, accessibilities. So this can affect the patient, their families, and other stuff. Major challenges remain. So how can we try to prevent it better, especially when we've got estimated number of cases by 2030 and 2050? How can we make more affordable treatments in worldwide? Because not every country has a free healthcare system. Okay. Is there treatments with less side effects? And this is why, as part of the World Healthcare Organization, their goal is to try and have a sustainable development goal to try and raise awareness in terms of the health and education and many other aspects to try and lower the rate of cancer. We will now progress on to what is World Cancer Research Day. So World Cancer Research Day is an organization that has been established in 2016. And its current theme this year is about cancer research needs us all. So the aim of this initiative is to raise the importance of cancer, try to develop better tests to detect cancer and new treatments. So their goal, so they mainly consist of 10 organizations. So they aim to unite the researchers, health organizations, educational institutions, and the general public about its importance. So I would strongly advise you to visit their website, www.worldcancerresearchday.org, to find out more about their work and what are they all about. So what we're going to do, we're going to pause. It's about with approximately an hour now. So we're going to pause the lecture, have a refreshment break. Okay. You may be free to continue if you wish. After the break, what we aim to do is to discuss what are the key steps involved in conducting research, what qualitative study is, and some examples. So I look forward to discussing this with you after the break. Hello, welcome back. So the key points so far within this lecture is that we discussed what cancer is. So cancer is the uncontrolled growth of abnormal cells. We tried to explain the difference between benign and metastatic. So with the benign, they remain in the original sites of where the cancer started. Metastatic, they actually grow away from the primary sites and try to affect the other organs and tissues. Genes and the lifestyle factors, they are the main risk factors of cancer. We also discussed why is cancer research important. There's a number of cases that has been increasing, and also the death rate is increasing too. The number of money that is being, or the amount of money that is being spent on cancer care around the world is increasing, and cancer research can be done in the laboratory. Surveys and monitoring how many people have had cancers or passed away, and there's other methods too. So now what we're going to do is we're going to start off by discussing what are the key steps involved in conducting research. So there's a number of different steps involved when we want to conduct research. The first is the aim. Why are you doing this research? What's your goal? Okay, so you have a start point, you have an end point. What's the end point? What are you trying to achieve? Now, the method is the planning. So you need to think of a way of how you're going to design to be able to collect the relevant information ethically. So if you're going to be collecting patient data, how are you going to do this? What information are you going to be collecting? Why is this going to help you for your research? If you're going to be doing, for instance, a laboratory, okay, what is your experiment about? Has somebody done this before? Has somebody not done this before? Have they done this similar? What results have they found? What results are you going to get that's going to make it a difference? Okay, so this is why it's very key to know what you're doing. Because if you don't have a name or knowing what you're doing, how you're doing it, you're a bit lost within the research. Okay, so this is why the initial and the most important step is to aim and the method. Now, once you get the results, you need to utilize maths. Why? Because as we looked in those previous examples earlier, we saw some numbers, we saw some graphs, and we saw some percentages. That's what statistics is about. It's utilizing maths or different methods using maths to try and understand what the data is trying to show you. Okay, what is it telling you? Is there an increase? Is there a decrease? Or is there no response? Evaluating the data. So this is important because you need some kind of comparison to understand how your results stand out. What has the other research study achieved, or is it similar? Is it different? Why is it different? What is it suggesting? What is it? How is it? What is it telling you? Okay. The conclusion is summarizing. So here's my data. here's what the result's telling me. Here's what, after comparing it, this with other researchers, this is what it's showing me. So what's the end point? Okay, how will it help? Is this the end point of this cancer research study, or can I do further investigations as a follow-up step to find out, okay, if I change this uh, this drug here and combine it with another drug and see its effect, if I change the dose here, or if you utilize a different cancer cell, if I try to improve my survey questions, or increase the number of sampling, or so forth, or the method in how I'm giving the surveys. So this is the key steps important in trying to do research. So what are the different types of research? There are so many, and I mean so many different types of research, and it all depends on what's your aim and your subject area. So within cancer research itself, there's different types, and research generally can be found with every subject area, from engineering to arts to humanities to languages to many, many different types. But in relevance, in terms of the cancer research field, within that itself, it depends on what's your aim, what type of cancer research study that you want to do. The main two types of research that can be found within cancer research and also in generically with other research subject fields is quantitative and qualitative. So our goal here is to focus on cancer research. So understanding what are the different types of cancer research related topics that are associated with qualitative, and similar to case, apply the same principle with quantitative. So now we're going to begin with qualitative. So what is this qualitative? Is it quality and then ative research? What is it associated with this? Qualitative is what you see, okay? What you see, the words being used, perceptions, interpretations. So, for instance, the feelings that has been said. So for cancer patients, their perceptions, it can also be healthcare professionals. The words utilized, the interpretations on their faces, all of this relevance undergo in the category of qualitative research. And within a cancer research study, you could invite somebody who doesn't have a cancer. So you could compare cancer patients with non-cancer patients and understand their awareness about cancer. So what are the features of qualitative research? So it can be subjective, so feelings, their experiences during the cancer journey, and it can involve open-ended questions rather than having yes, no, or don't know, or rather than having options that state, for instance, what do you think is the main risk factor of cancer? And then you need to select an option of this. It normally consists of one question and several lines where the participant who is enrolled into the study is able to express how they feel or the experience during the treatment journey. It can be also not only within a survey or a questionnaire, it can also be done within focus groups. So you could have small group discussions with an approved interviewer. and she'll be asking some interview questions which you can participate. This can be done face to face, on the phone, or written, and this provides in-depth research on topics that are not well understood. Because when you have closed-ended responses, which is yes, no, don't know, or you're not really you're collecting kind of like key information, but you're probably missing some insights that you may not well understood, because they'll be utilizing each cancer patient or cancer healthcare professional, anybody within who's enrolled within in the cancer research project, and you're asking them a question. They may utilize different terms, and these terms may interpret different meanings about the cancer experiences on their journey. And in terms of generically, it can be utilized within the cancer research. It can also be utilized in other subjects, such as those who are in politics or social work, psychology, which is the study of the human mind, anthropology, which is the study of the human behavior, culture, and society, law, and education. So there's a number of fields of it that can also utilize qualitative research. So can anybody just create a survey or ask a cancer patient about the experience just like that? Or is there prerequisites or steps that needs to be done before the research? So this is what we're going to be discussing now. And yes, there are prerequisites. The first is the whole research study needs to be ethically approved by the Law and Research Committee. You won't be able to, for instance, to collect blood samples or urine samples or any bodily fluid samples from cancer patients without their approval, to state that they don't mind that their samples are used in research. Okay. Number two, not only just the cancer patients, but within the research council or the research committee, they need to ensure that the method of how you're actually contacting the target audience, how you're collecting the data, and how the data will be used, and managing risks and making sure that there's anonymity utilized in this research study is actually being applied. Because what you need to do is ensure that you need to protect the patient data. You need to ensure that they're not revealed. They need to be aware of what the data will be utilized for, how is it going to help, how you're going to be managing risks, and so forth. So this is what project proposal is about. It summarizes how many people you want to get involved, how you're going to contact them, what's the method, is there a consent form, is there a participation information form, who's going to be involved in terms of this project proposal, where is it taking place. If you're doing a non-laboratory, which is a survey, it needs to be an approved, confident location. At a certain time, an appointment's been made, everything professional, okay, for it to be conducted. Because what you can't do is to collect data of anything or anybody enrolled in a research that they don't know that they were enrolled in a research study, or that their data is being collected and shared without their approval. So this is why everything needs to be ethically done and approved. Now, we've mentioned earlier that there's a research study information worksheet. So What that does, it presents an overview of what the research study is about. Okay, so it's not technical terms or so forth. It's just necessarily, okay, what this research is about, what particular type of cancer, how is this research going to help cancer patients or those within the cancer profession, and also the formal consent, because what you have tendency is that one would say, okay, I've had this information, I'm aware about it, but I don't remember signing a consent form to say that I want my data to be collected, or I want my bodily sample to be utilized within a clinical trial or a clinical study. So this is why it's very key that you have a formal consent form that is being signed, that you have a copy, the participant has a copy, and they are aware of what the study is about. And within, for instance, like a clinical trial or any part of the study, they have the right to leave at any time point of the research study. So this could be in terms of tense, stress, or other contributing factors that have led them to say that, look, initially I was interested in partaking in this study or this research project or training project. However, I'm no longer interested into continuing this because of ABC, and therefore I would like to leave this clinical research study. So this is the case there. So this is why you need to have a conscious intention with the intended outcome. And there's also another reason why, for instance, when you have a clinical trials, you have a certain number that are enrolled and participated. But by the endpoints, that same number does not exist. Either they have, for instance, failed during the cancer clinical trial, for instance, of responses, or within the criteria during the cancer clinical study, or to the fact that they've did not want to continue due to other factors, and so this is needs to be clearly aware. With interviews or focus groups, this needs to be within a location, time, and date that has been approved. Okay, so a formal invitation, formal meeting, and so forth all needs to be conducted appropriately. So this is an example of a qualitative research within cancer. So, for instance, in a cancer focus group, a group of volunteers who opted to partake in a small group discussion, they have an arranged time, dates, confidential location with an approved interviewer. Example of an open-ended question would be, how did you feel when you were initially being told that you had cancer? So when you have those few lines, it allows you to express, okay, rather than a limited yes or no or other options. And also, there's some factors that you may want to consider that can affect the response. This is age, race, gender, ethnicity, other health conditions, socioeconomic status, whether they have dependents such as children or care for somebody, their aspects in life in general, and how they handle problems and other factors. So what are the main types of qualitative research? So earlier on, we've given a description or defined what are the features. Now, within the qualitative research, there are different types. So we're going to be discussing. So there are different types of qualitative research. So we have ethnography, focus groups, content analysis, narrative, phenomenological, grounded theory, and case study. What we're going to do now is ethnography. So what is ethnography? So ethnography is to understand how culture, tradition, or belief can affect the perspective on life. Okay, so why is this relevant for cancer research? So your target audience could be patients who actually live in the countryside or the village with those who live in the city. So you may want to compare those two types of patient groups on their accessibility to cancer care, um, how available the health information is, and their way of life. So, for instance, you may want to compare how they live with the disease, explore cultural factors, compare different countries, the cultural factors, and how understand how they actually live. And this will help to get a better understanding, to observe the way of the life, um, more than interviews and surveys. The next type we're going to be focusing on is focus groups. So earlier we discussed what is focus groups. So this is a small group. So just a gentle reminder, it's a small group of people that have an in-depth discussion on social issues. What's the relevance with cancer research? Well, the target audience could be cancer patients. You may want to discuss what are the viewpoints of the cancer patients on how the government is supporting the economical burden of cancer care. Okay, what are they doing to lower the health costs? Content analysis. So content analysis may involve analyzing or viewing a speech made by, for instance, a health minister or a healthcare professional or a patient. And you may want to look at, okay, what's been said, what's documents been shared, what images has been shared. So you're analyzing the content there. So you may want to, for instance, compare, for example, the facial expression of a cancer patient before or after procedure, what information that has been said by the health minister or the healthcare professional, um, uh, you know, what have they mentioned regards to it. So this is content. We will now progress with narrative. So this is a long-term project. So it may involve one or two areas overlapping one another. It could be in a form of interviews, so you're asking questions, documents, and themes. An example related to cancer research would be, okay, a journey of developing a device, so a technology device for cancer. So you may want to ask cancer patients their opinions, current cancer researchers, viewing documents, for instance, the protocol, patents, how is it being used, is it safe to use, and so forth. Another example would be, you may want to target cancer patients. So the initial signs and symptoms and the cancer journey, being able to identify how they actually feel and how it exists with their experience and identity. So you have multiple factors here, signs and symptoms, feelings, experience, and identity. So these are a number of subjects that overlap with one another. And this helps researchers understand the importance of patient-centered care. So you're not just looking at the disease there, you're looking at factors that are affecting the patient itself, that could be internal, external, and how to improve medical practice. So this could be associated with communication, clarity, being involved in decision making. There's a number of different factors that can affect different cancer patients. Phenomenological. So let's see what phenomenological is about. Phenomenological, this is a research that is trying to understand an event or miracle. It may involve interviewing documents or visiting places to get clear idea, okay, or clear data. So again, it may involve cancer patients. But what distinguishes, or what's the difference between phenomenological and other researches that we've mentioned earlier? Well, patient can be interviewed with questions on how this disease has affected their mind, okay? Or how has it changed the perspective on hope? Because hope means something to cancer patients, hope for a cure, a hope to be cancer free, a hope for a test to be clear, okay? The anxiety and the depression, for instance, of waiting for the results or going through the actual test itself, or through the surgery or radiotherapy, chemotherapy, especially when you haven't had this condition earlier. This is like first time of having being diagnosed by cancer, and this whole fear of mind and how it's changing your life adjustments. And you're just like, okay, this is the reality. This is happening. I'm gonna get through this. So the different responses of the psychological perspective on hope varies from patient to patient. It depends on the levels of resilience, how strong their relationship with the friends and the family, being able to fight all the way till the end, and having the will to live, especially with the new clinical trials. Keep trying, testing, wait for results. Oh, it didn't work. Let's try again. Let's find something else. Let's, try different treatments. And with this type of technique of having new clinical trials and hope and will to live, it helps to stop getting negative emotions or a negative mental state, and to see the world more positively. And it's not only just regard to the patient, but also the patient family, the patient's friends, the patient's the community, the healthcare professionals, because healthcare professionals, like, they see the patients day in, day out, okay? They see the amount of suffering the patient undergoes, and at the end of the day, they're a human being. They've got feelings. They want to see the cancer patients getting better. You want to see the cancer patients going back to their lives of how it was before, or try to improve the quality of their life as much as possible. And they're utilizing their knowledge, their experience, trial and improvements in order to do this with the different cancer care plans. And this is necessary. That it's very critical to mention this, that the healthcare professionals, they are human before they are a doctor. So they understand the importance of, having goals, having hope and everything. And this is why when we talk about medicine, it's about passion, it's about understanding the patients and doing that the best that you can. But it's very important that to understand that the doctors, they are humans, the nurses, they are humans, the cancer secretaries, or anybody who is involved within the cancer care plan, they are humans, they have feelings, they understand, they want to help. So it's very key to understand the importance of emotion, how it not only just affects the patients and the families, but it's also the healthcare professionals and anybody who's actually involved within the cancer care. Moving on to grounded theory. Grounded theory, this is a research that has a larger sample size. So that approximately is between 20 to 60 people. And it explores factors that can affect an event. So this also involves interviews and documents. So you may have noticed there's all of these qualitative research types that we have discussed so far, inclusive interviews, documents, surveys, questionnaires, but the method is different. So in how they utilize the results, or what's their goal of doing this. And Straws and Corbett in 2012 defined it as theory derived from data collected systematically and through a research process in which data collection and the analysis and theory that will arise from them are closely related to each other. So an example of this would be understanding patient decision making. What factors influences a cancer patient? So this may be associated or what are the preferences of cancer patients in their care? Okay, what do they want to, so when you give them a choice, what do they prefer? Do they prefer having a decision being made for them? Or do they like to be involved within their decision on, for instance, what tests to be done, what treatment to have, and so forth alongside their doctor? Okay, so if one is if a patient is interested in having a doctor making the decision throughout the treatments, this is more kind of like assessing in terms of their values and preferences. Okay. Now, and it's also all about the response of the physician. Does the physician understand or adhere to what the patient's decision is? Okay, to either stick to the treatment and improve the quality of life. What factors or such as stress, fatigue, aches, emotion, how do these factors influence the patient's decision on whether to do a treatment like surgery, which is very invasive, or would they like to do something else? Okay. Other factors you may want to consider is uncertainty of getting it again, so relapse, or for the cancer to actually continue growing, so that's metastasis. So all of these factors are dependent on the personality of the doctor, personality of the patient. Are they empathic? Are they compassionate? Do they kind of like understand and relate how the patient actually feels? Or are they kind of like robotic in their method? Or just like, hey, this, and so forth? Or are they actually kind of acknowledging the patient's feelings to say that, look, I understand that this cancer treatment is affecting you, but here are the other options, and we're going to try these options to see how it's going to help. Okay, so other factors may want to consider is a type of healthcare system. That's very key because globally there's different types of healthcare systems, so the approach or method or guidelines varies. And how accessible are they to the information that they receive. Last but not least is case study. So case study is like an in-depth data that can vary from on a subject from one person to actually examining to a country level. So you may want to explore different factors, or you want to use data to explain certain factors. So you could use data that examines different factors on a country level or on one person level. So this is our second break. I would like to pause the lecture. I hope you have enjoyed the lecture so far. After the lecture, we are going to discuss quantitative research with examples. Thank you. Welcome back, everybody. So the key points so far, which we have discussed together, is that there are two different types of research, qualitative and quantitative. Before research can take place, the study needs to be approved and signed by the subject, that they understand what the research is about and fully consent that their data to be collected and analysed. In terms of qualitative research, the interviewer provides open-ended questions, so this helps to dig deep into understanding how people feel, their experiences and perspective on the cancer journey. So this may mean to actually observe how participants live, ethnography, to understand how their culture influences the perspective. It could be interviews, focus groups, analysing documents and speeches. We will now progress on to introduction to quantitative research, where we will be able to describe, understand the different types and examples. Quantitative research, as the name suggests, quantity, numbers, how much, so it's all about numbers. What are the features of quantitative study? Quantitative research is where data is being collected and analysed using statistics and graphs. Amongst the examples of graphs as shown are column, bar, pie, area, line, and column and line. So when you have continuous data, line graph is most suitable. But if you have categorical data, where it's not something that is continuous, it's more categorical, such as colors or types of cars or age groups, then column and bar is more suitable. And there are specific types of subjects where quantitative research can apply, mainly medicine, science, psychology, sociology, and economics. So economics is all about money, sociology is the study of people, psychology is study of the mind, and then sciences could be biology, chemistry, physics, and medicine. It consists of combinations of these sciences, but also it involves in terms of the drug treatment, patient care, cancer clinical trials. We're going to be discussing some of this later. Quantitative research may occur in the laboratory experiments, case studies, tests, assessments, surveys, closed questionnaires in person, online, or via the phone. On the number of people that are enrolled within the quantitative research is a lot larger than qualitative. So the sample or the number of people enrolled within the study is a lot larger. And this helps to identify relationships like what's the cause, what's the effect, patterns and trends, being able to find these results and compare it or generalize it to even larger populations. So let's just say, for instance, you compared males and females in terms of their response to cancer treatment for a particular type of cancer, and you explored, for instance, the people of Birmingham. So based on the people of Birmingham, you may can generalize, can even look at broader populations. So rather than just looking at Birmingham only, you could look at the whole of West Midlands, or the comparing West Midlands with East Midlands. So you can generally develop the larger and broader populations there. You can also test the hypothesis, so prediction. So what do you think is going to happen? So let's just say with the case of the conjugates, as an example earlier, they utilized different concentrations of the conjugate and then compared it with the control and the solvent. So the hypothesis could be behind the concentration, the more cancer cells that will die. So the lower the percentage cell viability. So that could be a hypothesis or a prediction that you think may occur. So what's the difference between primary and secondary data? So within quantitative research, there's a big difference between primary and secondary. With primary data, it's the data that you have developed, you have designed, you have found based on the laboratory experiment. Or it could be the results from a closed-ended questionnaire which you have designed and developed and collected that data. So this is known as primary research study. But what's a secondary research study or secondary data being collected is already data that has been established. So this could be public database or other people's database, governmental agencies or research organization or academic institutes. And you're utilizing their data to analyze it and compare it with other people's data. Okay, so this helps to identify patterns and relationships. So, for instance, if you may want to collect the number of incidents that are worldwide, so you may go utilize the database that is published by World Health Organization, compared a country that is low income country with a country that is high income country or highly developed, and then think about what impact does that have in terms of the number of incidents in cancer. Okay, so this is secondary data. What are the variables that affect a research study? Let's understand what is a variable. Now, vary means change, okay? But when we refer to variable, it's talking about feature or a factor. So the dependent variable or the dependent factor is what is being measured. Control is what you're keeping the same. Independence is what being changed, and the confounding. So if we utilize the example for the conjugate, the conjugate experiment could be utilizing uh, uh, what is being measured, the cell viability, so the number of cells of cancer cells that have been remained alive after treatments, with just the control, the solvents, and the different concentrations of the conjugate. The control is what you're keeping the same. So this could be the location, the condition of where the cancer cells. So when you're actually counting the cancer cells, are you leaving it with the treatment for a particular time, specific temperature, location, before you're counting how many cells that's alive, and also the method of how you're counting the cells. Are you, for instance, utilizing a cell counter? Are you utilizing a laboratory test known as MTT assay? What is the method and how you're collecting the cells, or are you kind of under the microscope? So this is the method. Independent, what is being changed? So what's the factors that are being changed? In that experiment, they've used different concentrations of the conjugate. They've used 12 different conjugates. They've also used 4 different types of cancer cells. So these are some of the factors that have been changed within that particular experiment. So this is a case over here. If one is doing a survey, a close-ended survey, what is being measured? So for instance, you did a close-ended survey on what are people's opinions on a particular medication or chemotherapy, particular chemotherapy. So you're measuring how many people said yes or no, and frequency and so forth. Control is the number of cancer patients from a particular hospital or a particular age group, particular race, or what are you keeping the same? Also the same questionnaire, the same consent form, the same method in how you're conducting the study. What is being changed? So you're not utilizing just one patient, you could be utilizing many different patients. You may consider, for instance, are you comparing cancer patients with non-cancer patients? Are you comparing different races to understand the cultural awareness point of view? Are you comparing people from different faiths and no faiths to understand their viewpoints towards what is hope? So what you're actually changing. So this is some factor you may want to consider in terms of types of variables. What is a confounding? So a confounding variable are factors that affect the independent, what you're changing, and the dependent, what you're measuring. So if there is a factor or a variable that can affect both of these factors, that's called a confounding variable. And this ends up leading to misleading conclusions. So let's look at an example related to cancer research. So you conducted a study to determine the effect of exercise on improving the clinical outcomes in cancer patients. And it does not take into account how much level of knowledge these participants have, nor their physical activity level. So the confounding variable here is the previous physical activity and knowledge. Why? Because if they have previous experience within physical activity, they know what their level of activity is like, what exercises are good, how fit they are, how much the exercises that they do, when, where, and the knowledge. So the benefits of doing exercise, how is it going to help for cancer and non-cancer patients? going to the gym and making it routine. So having these previous knowledge or experience, or being within the field of this, or knowing someone who um, has this, can actually has this form of experience or knowledge, can actually influence their ability or their responses, and also their motivation or interest or understanding. Okay, so here is a physical activity pyramid from the dietitian resource. And for instance, level 1 is an everyday physical activity. You may end up going up the stairs, taking your pet for a walk, or you're probably just doing sprint walking. Then for stage 2, which is like dark green color, it's aerobic or recreational. So you may be utilizing some small stands, moderate to vigorous step, like a step, going up and down a step 3 to 5 times a week, 150 to 300 minutes per week. You may be cycling, scooter, ice skating, or running. Then you have the level 3 stage. So with the level 3 stage, it could be leisure, so you could do 2 to 3 times per week. So that's low to moderate. Time varies. So this could be, as the image presents, somebody who does golf or badminton or tennis. It could be strength related, so you could be 2 to 3 times per week. You know, with your dumbbells and trying to increase your muscle, strength, stamina. So this may involve 2 to 3 sets of 8 to 12 reps. It could involve some flexibility, so it could be yoga, stretching, press ups, so where you can hold for 10 to 30 seconds, 3 to 7 times per week. And then you would have sedentary, which is stage 4, which is limit sedentary time, which is like sitting on a chair. So there's different levels of physical activity, and it varies from person to. What are the different types of quantitative research? So there's several different types: descriptive, correlation, causal comparative, experimental, and clinical trials. We'll begin with descriptive. Descriptive research is being able to describe and identify characteristics of a population being studied. So, for example, you may want to investigate which age group is most at risk of a particular cancer. And for instance, if you plot this as a graph, the most likely type of graph that would be plotted is a bar chart or a column chart because it's categorical data. So on the horizontal axis, on the x-axis, you would have the different age groups. So rather than having 1, 2, 3, 4, 5, 6, all the way up to 100, and then more than 100, what you would do is put them into groups. So you would say, for instance, 0 to 20, 20 to 40, 40 to 60, 60 to 80, and then 80 to 100. Okay, so it's always ensure that the intervals are equal. And you would then, for instance, you may want to investigate how many people are at risk of a particular cancer. So let's look at for a random type of cancer, such as bladder cancer. Okay, so with bladder cancer, you want to investigate which age group is most at risk of a particular cancer. So you would have a table that has a list of patients, not their names or anything as such, but it would be anonymous. So it'd be one, patient 2, patient 3, or patient A, patient B, patient C. And they would have the age group next to it. And also whether they have cancer or whether they don't have cancer. And then you would count how many people that fit within each of the age interval group, and then you would plot your bar chart. So for example, you had, based on the raw data, you had about 25 people that were aged 40 to 60 that had bladder cancer. So you would plot that number on the actual graph. How many people had bladder cancer? What was 0 to 20? 0. So you'd put 0. So you wouldn't really have a line because it's actually on the 0 mark. If you had somebody between 20 to 40, and there was about 10 people from that raw data that were diagnosed with bladder cancer. So this would be mark it up to the 10, and how you would plot this. So this is how you would plot the data. So here is another example of a descriptive research. We are presented here with a graph by the source called SEER, and it's collecting data from the years 2016 to 2020 of how many cases there are of esophageal cancer. Esophageal cancer, otherwise known as throat cancer or the food pipe. Okay, and it includes all races and also both sexes. And on the x-axis, we have the different age groups. So you have less than 20, 20 to 34, 35 to 44, 45 to 54, 55 to 64, 65 to 74, 75 to 84, and then you would have above 84. On the y-axis, or the vertical, or the line that is actually standing, is the percent of new cases, and that's going up in fives. So the purpose of this data being shared is to present how descriptive research can be illustrated. So with less than 20, there is no cases, hence there's a 0 mark. With 20 to 34 years of age, there was 0.4% of cases of throat cancer. Between 35 to 44, there was 1.8%, so there's a slight increase there. And between 45 to 54, it moved from 1.8 to 8.3%. And then from 45 to 54 to 55.64, there was a massive increase. So it went from 8.3% to 26.5%. And then even higher number of cases was found in people aged 65 to 74, because it went from 26.5 to 33.8. And this is why the median age at diagnosis is 68, because it has the most cases within that bar, and it's the highest from all of the bars presented. However, when you look at 75 to 84, the number of cases dropped down, so it's 20.8%, and then it's much, much lower for anyone who's above 84, where cases are 8.3%. So this is why the median age at diagnosis is 68, and the most frequent age group that are at most risk of throat cancer is 65 to 74. Correlation. Correlative research. So this is describing two factors to determine whether there is a link or relationship between them. So in the previous example, we have categorical data such as age groups. However, when we refer to correlative research, it would need to be more of comparing two factors. So it could be the number of cases over a period of time, and this is known as continuous data. Other examples of how correlation can be presented is called scatter graph. So scatter graph is, let's just say, you're comparing the marks of a classroom, of how many people, what grade did they achieve, for instance, in one subject, and comparing it with another subject. Yeah, so this is considered as and then you would need to have a line of best fit, and you put a line through it. So this is another example of correlation utilized, and we can see this over here. So this data is from Cancer Research UK, and it presents new cancer cases per year for all ages combined, all cancers excluding, so not including, non-melanoma skin cancer. This is a particular type of cancer that is not included, but they're referring to all types of cancers. So in the pink there is both genders. In the blue, it's the men, and in the dark blue is the women. Now, at the beginning between 1993 to 1995, the number of cases for all persons was 257,000. Now, with the men and the women, you won't kind of see a significant difference during that year, but it's approximately about, let me see, about 110,000, possibly, new cancer cases per year. And then there's been an increase by 50% in 2017 to 2019 for both genders, where there's 384,000. And as for the men and the women, what you can see over here is there's not much significant difference to both of the gender, but you can see it's steadily increasing from 1998 to 2000, and 2003 to 2005, still consistently increasing on both sides. Then there's a slight overtake over here with the male incidence, and then approximately 2018 to 2020, you can see there's some slight increased difference of the male incidence. So this is with the male incidence more 200,000 in 2018 to 2020, but there's a slight decrease just under it for the female incidence. And then from 2017 to 2019, it's been estimated that by 2038, 2040, that there will be a 32% increase, so 406,000 Okay, so that's a 32% increase from 384,000. Okay. And the men cancer incidence is approximately about 200 and so if that's 257,000 over there, so approximately around 257,000 for the males there, and women just slightly below that. Yeah. The next step we're going to discuss is causal comparative. So causal comparative research, this aims to find out the cause and effect. So one factor is changed and is not manipulated, whereas the other factor or variable is manipulated. So, for example, you may want to compare the effect of one chemotherapy treatment with two groups. One receiving the treatment, so they've been exposed towards the treatment, whereas the other group is not receiving the treatment, so they're considered as the control. Example 2 would be if you wanted to observe patients on how they're doing with an older version and newer version of the same drug. So you want to consider, okay, this is a drug, it's made of this particular material. Now the newer version has been adapted and much more improved. So the effect is the difference in the drug and the cause is the use of a new drug. A reminder that transcription is when the base sequence of the DNA is being copied. So when it's being copied, it needs to the DNA needs to be one-stranded, so it needs to uncoil itself, copy the information as a single strand RNA, and then when the information is copied and into the messenger RNA, what happens is it travels to within the cell to a structure called ribosome, and ribosomes is where proteins are being made. So in the what happens is that the genetic code or the genetic information that is presented by messenger RNA is being translated. So there's no more genetic code information left. What it is, it's been translated into an amino acid, because with each amino acid, it has three genetic. It has like a genetic code, so it gets translated until the whole protein is being made. So one after, like the triplet code, one after the other, one after the other, until the whole genetic sequence has been translated, and that is what makes up the protein. Okay, so initially you have the DNA. It's a double-stranded ladder of genetic information. That gets uncoiled or untwisted to, and then the information gets copied into the messenger RNA, which is like a single strand. Then this messenger RNA travels to a structure called ribosome inside the cell where the proteins are being made, and it translates this information that the messenger RNA is bringing and is converting it into an amino acid. So these are like the building blocks or the bricks that make. Now experimental research does involve cause and effect, but there's a number of factors that are being measured. So one example is a group of researchers did a research study on mice as tumor models for bone, skin, and brain cancers. Now, let's pause over here. You may think, oh, okay, why are they using mice as tumor models for bone, skin, and brain cancers? I mean, the reason being is we cannot just utilize any type of tested material and use it straight away onto the human subject without knowing whether it's safe for use, whether it's what dose kind of is safe enough for human use. Because when you with some medication, when you use very, very high levels, it gets really toxic. Um, and the quality, how effective is it? Is it just temporary, or is it consistent and it has long-term positive effects? And this is why tumor models such as mice are being actually utilized, where they can be tested upon. And mice were divided into two groups. So this is just an example of a study, and it was tested with different vaccine designs of the mRNA vaccine to determine whether it is safe and will give the highest effects before progressing onto human subjects. So with the mRNA vaccine, it, it targets specific tumors in order to activate the immune response, because it's flexible and can change its speed. So let's look at overleaf just to get a clear idea. So here we have the HPV, the human papilloma virus. So this cancer is more linked towards cervical cancer. And you can see it has a DNA genetic material inside. It also has like a protein coat called a capsid around, and it has also like structured proteins that are um like spikes just outside the human papilloma virus. So what happens is the mRNA, okay, um, of the and enters the cell, enters the human cell, and then what happens is the cell starts to make the proteins. What type of proteins are we referring to? We're referring to the proteins that make up the spikes outside outside the virus. Why are we making the spike proteins outside the virus? Because once the cell is being able to start and produce the production, it's able to recognize by the immune system more quickly, and therefore is able to combat it much more easier by producing antibodies. Yeah, so it can kill the cancer cells. So this is why you have the mRNA of the actual virus, so it can be entered in, make up the proteins, so your body able to recognize it better. And yeah, so this is why it's called an mRNA vaccine. So what we're going to do, we're going to pause the lecture and have a break. So after the break, we will discuss what a clinical trial is and particular types. Hi everybody, welcome back. I hope you enjoyed your break. So we're just going to have a quick review of what we've learned so far about quantitative research. So quantitative research is all about numbers, statistics, and graphs. The similarity, or what makes it similar between qualitative and quantitative, is that they both have surveys and questionnaires. However, the type of questions are different. In quantitative research, the questions are close-ended, so to provide concise responses. However, with qualitative research, you have more in-depth insights. Some types of quantitative research methods include descriptives. So how can data be described? It also includes correlation research, where you show relationship between two factors. Other examples or types of quantitative research are comparative causal and experimental methods. So this is where you're investigating multiple factors, more than one. The same method on ethical approval lies for both qualitative and quantitative. Why? Because in quantitative, when you are doing experiments, you may utilize human samples, human tissues, or their organs from their bodies. And this is why written consent is needed for it to be utilized in research and training. In some cases, when one is undergoing a operation, there's always potential risks. So they would ask you to sign to say, okay, if anything happens to you, what do you want to do with your organs? So this is why some would sign to say, yes, I understand, and this is how what I want to do with my organs. Some would choose to donate, and some would not want to donate. Okay, so this is why it's important to acknowledge the risks that are involved in the process. The next aspect we're going to go through is clinical trials. So let's do this. What is a clinical trial? So throughout the lecture, you may have heard me mentioning cancer clinical trials. We will now be able to provide an insight to what it is. So clinical trial is a type of experimental study, okay? And it researches the effect of drugs, researches the effects of devices, surgical procedures, or diagnostic procedures, or if there's a new technological device that is being used within a procedure, any other products before they can be formally utilized within health institutions. What are the different types of clinical trials? Now, according to Cancer Research UK, there's a variety of different types of clinical trials. We have prevention trial, so how effective is a treatment in preventing cancer? So you have some treatments that are, killing cancer. So, and there's some that is trying to prevent cancer. So this is commonly done if the patient has a family history or are at higher risk because of the lifestyle factors. So examples of preventative treatments could be weight loss or stopping smoking or alcohol, avoid chemicals that cause cancer, eating healthy diet, protecting your skin, so using sun cream, limited use of sunscreening and so forth, and being involved with cancer screenings and doing exercises. So these are like preventative measures, we call it. So which treatment or which preventative treatment is better, and so screening test. So this could be an imaging technique. So with biomarkers, so biomarkers are markers which you can test within the urine or within the blood or the tissue And when there's high levels of this protein per se, it suggests there's an increased risk of cancer. So, like with prostate cancer, you have PSA. With ovarian cancer, you have CA125. There's so many different types of cancers, especially you have biomarkers for bladder cancer, colon cancer, gastric cancers, so many different types of cancer. So it helps to detect those at risk. So it does not necessarily suggest that you do have the cancer, but having high levels of this protein biomarker suggests that it's an early sign, or you're most likely at risk rather than have the cancer. And require further investigation so the necessary cancer treatment can be applied. Now, with treatment trials, this may be associated with chemotherapy. So, cancer killing cells using chemicals, are they better alone or are they better combined? So, just like with the example of cisplatin and phenolphthalein, is the new drug better than the current drugs? How well did the patients respond? Okay. With the surgery, what are the survival rates by doing this, for instance, robotic surgery or using this particular device or tool? Is it effective? Is there less side effects? Which patients are suitable? Can it be done in the early stages of surgery, or can it be done in the later stages? So in most cases with surgery, the sooner you actually detect the cancer, and the smaller the cancer is, the more it's able to be extracted much more easier. However, the larger the cancer is, the more complicated the procedure is. So there's more risk of complications after surgery. And sometimes you may need a combination of different treatments. So you could utilize, for instance, chemotherapy to shrink the cancer, then the surgery to extract it. Sometimes you would need to have multiple sessions of chemotherapy and radiotherapy, because surgery is ineffective at a later stage. So it varies with cancer types, and this is why what type of surgery, what are the survival rates, and where can it be done in early or late. With radiotherapy, which dose is effective and causes minimal damage to normal cells. So you have two different types of radiotherapy. The external is when the radiotherapy is externally added to kill the cancer cells, and then internal beam is when it's internally, like when it goes inside to actually kill the cancer cells. And it varies per type of cancer. And the disadvantage of this case is that it not only destructs the the the, um, the cancer cells only, but also the normal cells. So this is how much minimal damage can it affect on the normal cells. And this is a similar case with the chemotherapy as well. And which dose is effective, because what you need to understand is that too much exposure of radiotherapy, or or radiation itself is a cancer causing issue. So this is why there's limited use, or how much uh, 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 cancer X-rays or gamma rays that are being utilized each year, even though there's a small percentage risk of this actually even occurring. But nevertheless, this is why dose is very key and is measured in gray. Now, with medical devices, how efficient is it working? So, is it used to detect cancer or another disease? How effective is it compared to other techniques? Quality life, longer lifespan, independence, mental health. These are lots of factors that one may consider with each of the modes of treatment. What are the main designs of clinical trials? So, there's a difference between type and design. So, even though you have preventative and treatment cancer clinical trials, within each of these types, there's different designs. Just like with vaccines, you have different designs and styles. Just with the cancer clinical trials, there's different designs and styles. And one of the main examples is randomized control trials. So random human participants, so they are being randomly selected to undergo the treatment or the procedure. So they could be randomly added to the control group who'd have no treatment, or is considered as a current gold standard treatment, and they have been allocated a random number generator. However, whether the participant has the treatment or not, what we consider as a control variable is the same number of participants in each group, where they have the baseline characteristics. So that could be maybe age, that could be gender, another format. This helps to lower the risk of confounding variables and control most biases. So if we look at this image over here, we've got 1 to 15 people within a population, but with the sample, so using a random number generator, you've got 2 chosen randomly, 5 chosen randomly, 9 chosen randomly, and 13 chosen randomly, and 14 chosen randomly. And there's no particular order. There's 2 and there's 5, and then there's 9, and then there's 13 and 14. So there isn't really like a sequence pattern or some form in how it's been collected. It's literally just a random number generator, and they're just into the treatment. Now, another type of or another design of clinical trial is open and non-randomized. So each participant is being recruited and is fully aware of the treatment that they will be receiving. So the methods are not considered as random. Placebo and control. So there's 2 sets of groups. Group 1, they will receive the treatment, so they're exposed to the treatment. Group 2, they will not receive the treatment, so they're under control. However, the human participants are unaware whether they're having the treatment or not. Okay. With the blind study, as the name suggests, there's someone that does not, the participant isn't able to see, and the number of participants, it varies per type of blind study. So when you have a single blind study, what that actually suggests is that the human participants is unaware nor have any form of information about the treatment that they are receiving. So yes, they enrolled with a consent form saying that, hey, I'm going to sign, I want to enroll in the study. Okay. I understand that this study is about, for instance, with ovarian cancer and so forth, but or another particular type of cancer, or they understand, just the gist, but they're not aware or have any information about the treatments that they are receiving. So they know that they're doing a cancer clinical trial for a particular cancer, but they're not aware or have information about the treatment that they're receiving. But the experimenters, because they are involved in the experiments, they are aware. So with the advantages is that it lowers bias and it increases validity of the results. Now, when you have a double-blind study, this suggests that neither the human participants nor the experimenter is aware of the information about the treatment they are receiving. Okay, so the experimenter may have like a kind of like a code of the drug, but they don't know what the drug is. Okay, so this is the question here. Now there's the triple blind study. So this suggests that neither the participant nor the experimenter nor the researcher who analyzes the data on the computer is aware about what type of treatment that they are receiving. So what factors affect clinical trial? The first factor that affects clinical trials is safety. So are there any side effects or risks? Is it minor? Is it major? How likely are they to occur? So this ensures to protect the public's health from harm. Quality, on the other hand, this is referring is how well is the medicine or product being produced? Is it long lasting? What type of material has been utilized? Is it according to the health and safety standards that they actually are meeting? Is there a particular level and potency for a metal or a medication that is being used for this? Are they of a better quality than the former projects? Is there a sign of improvement? So this is where quality comes along. Efficacy. So efficacy is finding the dose that gives the best health performance. How well is the product performing under controlled conditions? So this is the key, because what it is let's just say you've had two different cancer treatment concentrations. One was 50 and one was 100. There's not much significant difference between 50 and 100. So why would you increase it to 100 uh, uh, um, micromolar of a cancer treatment when 50 has a similar level? So um, this is about performing under control. What do you need to do before starting a clinical trial? So, besides the consent, of the study and information and being aware of the baseline statistics, the most important thing here is there's two key terms: pilot study, feasibility. With the pilot study, it's like a preliminary test just to ensure that everything's working well, and it helps to answer the research question. This can also apply even within the experimental stage, where you want to test it on a smaller level before utilizing it on a higher level. Feasibility, is it possible to do the study? Are the patients and clinicians happy to participate? And how long would it take to collect data and analyze information? So with every cancer research project, there's kind of like a deadline, and because they want to know how long will it take you to collect the data, analyze it, publish it, and so forth. What is a phase of a clinical trial? Phases are stages conducted, and they can be classified as early and late phase. So if it's an early phase, it means that it helps to determine if it is in a safe and if there's any side effects. But after so many experimental trials and so forth, you're now reached within the late phase. So this is where you're using a larger population to determine how effective it is compared to other treatments or a placebo. So placebo is like having a drug, but it doesn't contain the drug. So what are the phases of a clinical trial? The phases of the clinical trial consist of preclinical, phase 1, phase 2, phase 3, and phase 4. We will now begin with preclinical. Preclinical involves doing laboratory experiments to be able to find the right dose or concentration that is suitable and is safe to use on humans. This is why it can be done using cells or animals as models. If they are using cells or cell culture, this is known as in vitro study. However, if they are utilizing animals as models, it is known as in vivo study. Phase 0. So this is not commonly shown on flow diagrams. However, in some cancer clinical trials, it does exist. So this is where you are utilizing a low dose of treatment, okay, to determine if it is harmful. And they normally test 10 to 20 people. We will now progress with phase 1. In phase 1 clinical trial, the number of people in this phase is between 20 to 50 people. The aim is to determine which dose is suitable and if they experience any side effects. Phase 2. The number of people or participated within the cancer clinical trials is more than 100. Okay, so they're also trying to explore any side effects, working efficacy, and checking the best dose is actually consistent as the number of people involved in terms of the cancer clinical trials. The more the merrier. Phase 3. In phase 3 clinical trial, the number of people within this phase increases. So it's on a wider scale. It may involve people from different cities, different locations, and even possibly different countries. And amongst examples of what they commonly do at this stage is to test the drug and compare it with an approved or current drug. Or they could compare the drug with a placebo, which means a medication where there's nothing inside it. And this is used for evaluation. The similar factors of clinical trials are the same: effectiveness, quality, and safety. But this is more on a higher level because this is not looking at single use, but also whether the drug can be combined with other drugs. We will now progress with phase 4. So phase 4, there's diverse populations that is being used to monitor and its effectiveness, side effects, and safety. So, for instance, treatment versus control, no treatment. So what we're going to do, we're going to pause and have a break. Hopefully I'll see you after the break. Take care, everybody, and I'm hoping you're enjoying yourself. Hello, everybody. Welcome back. So, 2 points I was going to discuss today is in relation to clinical trials. So, a clinical trial is an experimental study used to investigate the treatment response. Okay, it can assess whether it's safe, is it effective, is it of a good quality. Some clinical trials are related to screening and prevention. So a clinical trial has different designs. So some involve humans being aware of it and some are not. Now, if they are human are aware of it, they are referred as randomized clinical trial. If they're not aware of it, it's considered as a blind study. So it could, if the person who does the experiment knows, but the participant doesn't, it's a single-blinded study. If neither the experiment nor the one who is participating in the study is aware, so that's considered as double-blinded study. However, when we include the researcher who actually analyzes the data, then this is referred as triple-blinded study. There's also different various stages or phases in a clinical trial, and it always begins in the laboratory to determine whether it's safe for human use. Now, during the clinical trials, the number of participants starts to increase, and determining the key factors, safety, effectiveness, and also of good quality, so where there's low side effects. And the treatments in the clinical trials are also compared with other approved options just to see if they are more effective. The first example of clinical trial is crossover. So a crossover is a type of control trial where each study participant will have both therapies. So they may start randomly to have treatment A. They may also, at the crossover points, begin to start treatment B. This is common if the outcome is reversible with time and if, for instance, symptoms may appear. Common cancers where crossover studies normally occur are prostate, breast, and gastrointestinal, so the intestines, tumors. So let's look at an example of a crossover clinical trial. So patients with previously untreated skin cancer, and the type of skin cancer we're referring to is a BRAF V600E mutation positive melanoma. So BRAF is part of a signaling or communication pathway for that helps cells to grow. However, when the BRAF is actually being mutated or there's a change in the gene, this is found in 50% of patients with advanced melanoma. And we can see the image here of how, for instance, when the BRAF has a normal gene, how it helps normal cell to actually grow. But when the BRAF is actually mutated, so it causes a loss of control in terms of the growth of the cancer. So this is the difference between them. Here is an example of a crossover study. So here are subjects that have been treated with two BRAF inhibitors. One is called dacarbazine, and the second is called dabrafenib. They vary in the median overall survival. So, what is the overall survival rate when having this medication? The overall survival rate for dabrafenib is 18.2 months. However, the overall survival for dacarbazine is slightly less, 15.6 months. So let's look at the flowchart to see how Latimer et al. 2015 have designed the breakthrough trial. So initially, in their investigation, they had 250 people enrolled. From the 250 people, 63 were assigned with dacarbazine and 187 were assigned with dabrafenib. However, unfortunately, those who had dacarbazine, some of them had disease progression, in other words, the cancer grew. So those were 36 patients that had to switch onto dabrafenib because of the disease progression. However, from the 63, the 27 did not switch to dabrafenib. They remained on dacarbazine. Why? Because their cancer did not progress. So there are different outcomes in each case. For those who stuck with dabrafenib throughout the study, so 187, patients that died was 78, patients that were censored was 109. However, when we refer to patients who continued to have dacarbazine, the patients who died were 8, and the patients who were censored were 19. However, for those who switched because of the disease progression, where they moved from the dacarbazine to dabrafenib, from those patients, there are 20 that passed away and there were 16 that were censored. So the 36 patients from the 63, so that's approximately 57%, had to switch because of the disease progression. But from those 36, most died, so they had 20, but 16 remained censored. The second example we're going to go of a clinical trial is cohort. So cohort is when data that have been obtained from groups of people over a long period of time has been exposed or not exposed to the particular new technology or factor of interest. And this helps determine what actually happens to them. And there's also a group where there's no allocation of exposure, and this is actually made by the researcher. So this is best to determine or study the effect of predictive risk factors. So just to break things further down, a group of participants with no cancer has information collected. They monitor who develops the cancer and who doesn't, and the participants who end up developing cancers are now monitored to see if they have any similarities for those of cancers who had before. So an example of a cohort is by a study conducted by Hurang et al. 2022, and they did a cohort study where they recruited 356 patients with colorectal cancer from January 2017 to December 2021. So what is the recruitment technique? So inclusion criteria are, these are the thumbs up. So if you have any of this, you're allowed inside the study. Exclusion criteria, there is a thumbs down. So if you have any of these are not recruited onto the cancer clinical study. So with the inclusion criteria, amongst the aspects that Hurang et al. Was searching for is that patients were first diagnosed with cancer. The confirmation was done by either with an endoscope. Okay, so this is like a thin tube instrument that is being passed through, and they can have some of the tissue being removed, such as biopsy. Now, patients who had colorectal cancer and grew to the liver metastasize, in other words, it initially was found within the gastrointestinal, okay, the intestines, and then they found some of the cancer cells affected the liver too, and they only underwent with CT or PET. So these are two of the most common types of techniques. So with the exclusion criteria, those patients who had undergone an operation, so those who had an operation, those who were treated already with chemotherapy, biological therapy, or immunotherapy, if they've had cancer metastasis, that grew to other organs other than the liver. So if there is growth of cancer, but it's not in the liver, then that's the exclusion. Also, they had metastases that were from other cancers to the colon or rectum, and they excluded long-term vegetarians or depended on folic acids. So initially, following the filtering through the criteria, the ultimate 259 patients with non-liver metastatic colorectal cancer and 97 patients who had liver metastatic colorectal cancer, altogether 259 plus 97 is 356 patients. So what despite they vary in terms of whether they have non-liver metastatic or whether they don't, they all 356 had some of the blood was collected 2 to 3 ml. They also combined the folate receptor NFR and labeled the circulating tumor cell together, and they were able to detect the levels of this protein called heat shock protein 90. What did the experimenters do next? So once they've combined FR plus CTC and HSP90 proteins, they measured it in both of the cohorts. Those with colorectal cancer and liver metastasis combined, and those with colorectal cancer with no liver metastasis. In addition, both cohorts had measurement of biomarkers such as CA199 and CEA, carcinoembryogenic antigen. Tumor immunohistochemical proteins such as P53, Pi67, CK7, CK20, V-catenin, villi, and S100. They also measured the tumor size and number of lymph nodes metastases. However, patients who did not have liver metastasis, they were seen every 3 months as a follow-up using CT or PET for any signs of liver metastasis. The researchers also created a mathematical equation to predict the risk of liver metastasis. So high-risk cohort of 159 patients and low-risk cohort was 197 patients. So based on these results, what they discovered is that those who have high levels of FR plus CTC and HSP 90 were at most risk of having liver metastasis. We will now progress on to case control. So with case control, patients are grouped into two: have certain outcome or disease, do not have the disease. So that's control. Now, similarities between the groups are referred, for instance, at the baseline characteristics like age, gender, should be the same for reliability. But factors actually investigated. So this is information that's been obtained on whether they have been exposed to the factor being investigated. So it has its positives, but a disadvantage with this is that people are asked and may think and remember when exposed to the risk factor, and not all information can be remembered accurately. So here is an image to help support your learning. So the number of cases divided between exposed, unexposed, and the number of controls also divided into exposed and unexposed. So this is in terms of the past, and at the onset of study and time as a factor. So one example is if one group consumes a particular food that increases the risk of cancer versus a group that doesn't. Another example is if you're investigating the effect of vitamin D. So is there a link between pancreatic cancer and vitamin D? So you have patients with pancreatic cancer and patients without pancreatic cancer. That's a control. So the exposure of interest we have over here is vitamin D intake. So all participants are asked whether they had vitamin D supplements to determine if it led to development of pancreatic cancer. So the strength of the dose and length of the time taken, it may also contribute or affect the cross-sectional. Cross-sectional. So this is a study that is performed within a particular population, one time point, or within a short period of time. And this is to determine if the risk factor has been developed, if there is any form of disease being present, and whether there is a link between the risk factor and the disease. So this can help determine how accurate is the diagnostic test, and if there is a link, what tendency that may occur is to do additional clinical trials, but of a different form, such as case control or cohort. So here we have an image that presents cross-sectional study, can compare groups within the same sample. So data is collected at one time point on one sample. Okay, so that's another example presented there. So example of a cross-sectional study is as follows. Example 1, there's prevalence of cancer in each UK region. Example 2, comparing patients who had a previous history of smoking and current diagnosis of lung cancer. Example 3, comparing patients in primary care using 2 criteria from the National Institute for Health and Care Excellence. So non-specific but concerning symptoms. This is NSCS. They would have site-specific alarm symptoms. So these are non-NSCS. So factors analyzed is socioeconomic status, whether that's a factor, disease characteristics to determine whether there's a link with long primary care intervals. Are there people with NSCS? Are they being diagnosed, for instance, within primary care, or are they actually being diagnosed at later stages or via the emergency presentation than non-NSCS? So do patients with NSCS have longer PCI than non-NSCS? So this is something one would like to explain. So what is a mixed method? A mixed method consists of qualitative and quantitative research. For example, through open questions, you may want to understand or consider how patients feel about their cancer treatments. And this is needed in order to be able to learn and gain an insight. So this is an example of a qualitative research to get an in-depth insight. However, a gentle reminder that when you develop a survey and it is now given on a larger scale, this is considered as quantitative research. So when they went in in-depth research to find out how patients feel about the cancer treatment, well, once they get the gist of it, they then create a survey that has closed-ended questions, and this is what's been given in a larger scale. In the second example here, you may create a survey to understand or find out the number of lifestyle factors preferred by cancer patients and figure trend over the years. So this example may involve a quantitative research, for instance, to find out what type of lifestyle factors that they do, how often do they do it, is it so you could do like kind of like a scale, Likert scale, how frequent is it, and so forth. And then later on, you may want to do a face-to-face interview to get an in-depth insight to find out the cause of why those lifestyle factors have been selected, okay? Or to find out from these participants, what do they think are the reasons why there's an increase or a decrease on the number of cases over the years. We will now review the qualities of quantitative and qualitative. So let's review the differences between qualitative and quantitative. So with qualitative, it involves subjective viewpoints. So this could be feelings, opinions, experiences, behavior, and perspectives. On the other hand, with quantitative data, it's more objective, it's factual information that's been collected. The type of responses that are found in qualitative data can sometimes be biased, and it can be influenced by the expertise of the researchers And this is why it can affect the reliability and how strength and valid the results are. And perhaps it may also lead to misinterpretation if their results lacks any form of cultural or contextual understanding. On the other hand, when we refer to quantitative data, because the information is objective and the feelings are absent, this lowers the risk of bias and increases the strength of these results. Why? Because when data is being collected, okay, it's converted within to the math formulas and tables and graphs in order to interpret the data. With qualitative data, another difference is that the sample population is small. However, with quantitative data, there is a large sample population. So there's lots of people enrolled onto the study. With qualitative data, it's intense. Similarly, with quantitative data, it is also intense. Why? Because with quantitative data, you may do an experiment. The pilot study might work, the pilot study may not work. You may, during the main investigation, that you have designed an experiment, but after so many hours, you find out that the result is not what is expected, okay? Or the experimental design has been failed. Well, that's not the end of the day. Why? Because you can try again and then design it differently and see whether you're able to achieve the aims and objectives of your experimental study. Another amongst the differences between qualitative and quantitative, with qualitative, you're having these interviews and questions being asked within a safe and confidential location. So this is where you've been professionally appointments being arranged. So that it requires resources, planning, and a committed team. Alternatively, with quantitative data, it's actually a lot more. Why? Because it involves a lot of laboratory chemicals. It could be software licensing, the apparatus, devices needed to utilize. So there's different aspects here. With flexibility, qualitative is flexible. It can help researchers adapt to context changes and explore. However, subjective interpretation actually limits flexibility. Alternatively, if we refer to quantitative data, it's a lot more flexible than qualitative because the type of data that's been collected and analyzed, and this is amongst the reasons, and it also helps test a hypothesis. Now, contextual analysis. Yes, there's involved with videos, images, recordings to provide an in-depth insight, and quantitative does no. There's lack of depth, and one may overlook contextual factors. Now, with surveys and questionnaires, there's open-ended questions and they have unstructured questions being asked. But with quantitative, it's structured, it's close-ended questions. So with the hypothesis, you would need a hypothesis in both of these cases, generate a theory. The last factor we will try to compare between qualitative and quantitative data is generalizability. So within qualitative data, there is limited generalizability because of the small sample size. However, because of the large sample size that is present in a quantitative research, This increases the level of generalizability because of its broader population. We will now begin within the last phase, which is how can the general public be involved in cancer research? So, for those who want to get involved in terms of cancer research, such as volunteering, um, uh, organizing events to fundraise, and so forth, this is where you need to contact a fundraising team or the volunteer team for that particular cancer charity and ask how to get involved. However, if you want to get involved in clinical trials, then you would need to contact your doctor that you are interested in joining the clinical trial. So they will then look at, for instance, your clinical history, whether you'll be fit enough to actually do this clinical trial. Because some clinical trials are actually paid. What they want to do is actually pay for the clinical trial, but then you would leave halfway for many stress or other considerable causes. So this is why you need to be fully aware of why you want to dedicate yourself towards this clinical cancer trial. There's a variety of different ones that you can get involved depending on your interest. So you can visit National Institute of Health and Research, Cancer Research UK, Wake Research, and WHO International Clinical Trials Registry Platform, ICTRP. So all of these have significant amount of cancer clinical trials that you may want to explore. It's now time to wrap up. Sad but not least, let's try to conclude and review the main key points. The World Cancer Research Day 2025 theme is Cancer Research Needs Us All. So this is integral. This is a very important because everybody wants to make a difference towards cancer research. And we're always looking forward to be able to prosper, succeed in developing effective tests and better treatments with less side effects. And if the cancer researcher is involved in qualitative studies, they try to in-depthly understand the cancer patient's thoughts, experiences, and communication on their journey. So some researchers live with them to understand the impact of culture. Others just do interviews, questionnaires, small focus groups, and analyzing documents. But if the cancer researcher is involved in quantitative studies, they plan and design experiments that you can't do at home. So they analyze the data to try and make progress to understand what's happening, what the results mean, whether it should be go ahead to actually do it in human subjects. Additional take-home messages is that it's fundamental that to ensure research participants voluntarily participate and are aware of what the study is about and consent their data to be collected. Now, it's part of the theme of World Cancer Research Day. There's a hope that cancer researcher, cancer patient, and the families, they hope that tomorrow is a better day. So never, and I mean never, please underestimate the power of cancer research. We will now go through the references. So here are some of the references. For any information that you may have and would like to find out more, please do not hesitate to review these references. You can find a small citation in every page, and you can refer towards the end of the lecture to find out where these references are from to get the full reference. Or alternatively, you have the lecture slides, so the PDF, and you may be able to review the lecture notes as well as the audio together. I would like to thank you for listening to this audio lecture. I hope it may be of help for your personal understanding. I also hope it may be of a form of assistance for your series of coffee morning lectures, or if you organize a one-day workshop or several-day workshops, and it may be of assistance for your target audience. Please do inform us about your thoughts of this lecture by emailing us the Farah Saeed trust@hotmail.com. To find out more about FST, please do visit www.farahsaeed.org. We have also organized alternative projects as part of this great annual event. Amongst them are past, present, and future cancer researchers, where we'll be able to understand their contribution towards cancer research. Another project involves understanding the importance of cancer, what cancer research means to them, from people around the world. May I once again would like to thank you for listening. And to find out more about World Cancer Research Day, please visit their own website on www.worldcancerresearchday.com to find out more about their cause and support their cause where necessary. Good day to you all and take care.